And please feel free to drop your questions in the Q&A panel throughout the meeting. If you face any technical issues, you can reach out to us at sales@biologit.com. I'll put that email address in the chat boxes. We can start now. Thank you. Over to you, Nicole.
Hi, everyone, and thank you so much for joining the webinar in a Friday afternoon, and I hope you're well and enjoying the bit of sunshine. Today we're going to be talking about AI-powered global and local literature monitoring and what is new. I think a lot of people have seen my presentation before. I've given the presentation a couple of times, but this time we want to be more of a live environment where Bruno is going to do a demo and show all of the new things that we have been doing, and then also the roadmap for Biologit. Next, Bruno. So for anybody who doesn't know Biologit, can I need to mute the new people joining? Can you hear the beeps kind of back to you, or is it okay? Okay, so an introduction to Biologit. So who are we? So we are a global technology company. We are based in Ireland, and we have deep expertise in pharmacovigilance and engineering and AI. We work with pharma companies and CROs. We work with all sizes of company, from very, very small to large companies, and we drive to improve the methodology that people are using today for literature monitoring. And how do we do what we do? So we are compliant with the literature monitoring, and also very up to date with any required regulatory requirements. Next. So when we talk about AI, the first thing that comes into mind is, what do the regulators and how do pharma companies can make sure that the use of AI is compliant? So there are kind of a few steps that you need to follow. So to adopt quality risk-based approach, so that is one, and that is across all of the regulations and all of the benchmarks that have been put in place in the last few years. Is always the first thing that they say: adopt a quality risk-based approach. So if you are doing a process where it's very important and is putting patients at risk, for example, then obviously you need a lot more scrutiny in terms of making sure that it's doing the right thing. Human involvement, so oversight and human in the loop, and human supervising in the loop, or automated decisions with QC. So there's a lot of requirements in terms of human in the loop or human oversight to make sure that the decisions that the AI is doing, they are not causing any risks or unnecessary harm. And then there's also the performance transparency. So you need to make sure that what you're doing and how the performance of the AI is effective and reliable, and then that the systems that you're using are transparent in terms of showing the performance. So you can, within pharmacovigilance, decide if what you're doing is bringing the right results or not. For example, for the literature, if it is missing or not missing any important safety information. And then the last one is the life cycle maintenance. So clear documentation, communication about the process, traceability and validation. And that has to happen on an ongoing basis. You can't just do it at the beginning of the project and forget about it, but it has to happen within the life cycle of the product. Next. I wanted to find a new numbers to show this, but this is now it's quite old because it's going from 2022 and we are in 2026. But this very clearly highlights the need of automation for literature monitoring. And these are information coming from the EMA on the literature monitoring that they do internally. It shows how many articles have been reviewed. So this is all done by a human, I suppose, and their EMA is looking at implementing new technology and AI to their systems as well. But you see the number of articles reviewed increasing, and it goes from like 300 to 700, and the number of unique cases are not increasing. They are remaining the same. So you are reviewing a lot more articles, but the output of it is not increasing. So there's a huge manual and admin type of work that happens in the background to find valid ICSRs or signals and so on. And that is very resource and budget intensive and carries a lot of risk for errors and gaps. Next. And then we have the global literature, but then we also have the local literature, and that drives further complexity beyond volume. So there's a lack of standardization in local sources. So the local sources are all different. They could be a PDF, they could be in print, they could be a website on the internet, it could be a magazine style of a journal. So they are not as consistent as you would find with the global searches or the global information and sources that you find in databases like PubMed. You will find a very there's no standards for that. So they all come in different shapes and sizes. And then there's also challenges with accessing the local literature. So for some journals, you need to have a subscription. For some other journals, you need to be a specialized consultant to get access to that. For example, some others are in print and they only deliver it to the country where they are printed and so on. So the challenge to kind of automate the whole local literature is quite it can be difficult, but I think that the methodology of doing local literature can be changed, and we have changed to an automated methodology. And then you also have the barrier that the languages, so there's a number of languages around the globe, and then most of the local literature will be in the local language. And then there's also the regulatory expectations. So different countries are looking for slightly different journals, and maybe across the globe, there might be different regulatory timelines and so on. So the process needs to be incorporated to make sure that it can be harmonized within the whole process that the company is doing. Next. So we have come up with within our platform, we can look at the local literature, and that is just one example, and Bruno will show that a lot more in detail when he goes into the demo. So we have online journal, so anything that is open source, open access, we will collect that. And then there's anything that is subscription based. So this is a private client instance, for example, because the client is paying for the subscriptions, and it's only the client that will have access to that for copyright reasons. And then you have in print journals where there is also private instance for the client, where the in print journals are digitalized and ingested into the platform. So we ingest all of those sources. They are different sources. We ingest them all. Some of them, for example, like the global journals, they have an ISSN number, but some of the locals wouldn't have, because they're just different sources from different places. So we will give it a unique number and then digitalize and have it all into the database. So once it is all in a database, you can run a single query for all of your local literature, or you can run separate queries for all of the products that you have in different countries. So you can have individual searches or a a full global search, and it can produce country-wise results for assessment. And then that can be fully automated as well for your full assessment. And I think that will be a lot, like you can see it better once you see with the demo. Next. So why are we different, I think? I think we started working with data when we first started Biologit, and we started building an open source, open access database where we incorporate any sources that is available globally, and then we incorporated all of this data. And today I can say that we have the largest database in the world because we have data from all over the globe, and we have data from a number of different databases. And that database is helping us to have the data that is necessary to do the assessments for pharmacovigilance. And then with that, we have an AI that is proprietary to us, that is extracting the safety information from the text of all of the text that we have, which is sitting in the database. And then we have a workflow that is helping companies to do the processing of the literature and find if there is a valid ICSR, if there's a new safety signal for QC, for any of the activities that they need to do. So everything that they do within the literature monitoring gets done within one workflow, and people can interact with each other as well. So you can have an initial reviewer, you can have the scientists involved, you can have your quality control also involved. So it goes from the data all of the way to the integration with safety systems. So for example, today we integrate with a number of safety systems. We integrate with Viva, we integrate with Argus, we integrate with Argus Global. And our aim is to be able to integrate with any system, so to facilitate the flow of information, and especially information coming from the literature. Next. And also, what can you do with that data? Who wants data coming from the literature? So today we do it purely for pharmacovigilance. So pharmacovigilance is one area that we know that needs literature monitoring on a regular basis. Most companies will do it on a weekly basis, but also we also have other areas that need literature monitoring and that we could use the data for. So for example, for regulatory, for clinical, for epidemiology, for toxicology, and so on. There's a lot of different activities that need literature to improve and to give them the insight. So our database could possibly be used in the future for a lot of other instances where you need to collect information and use, for example, even if you want to use models on top of it to collect the necessary information from the database, from the data that we currently have. Also, not only pharmacovigilance, but medical devices, for example, they need to do literature monitoring. Animal health needs literature monitoring, and also nutraceuticals and cosmetics and so on. So there's a vast amount of data that can be used for a number of use cases. But today, within Biologit, most of the companies we work for are doing literature monitoring for pharmacovigilance or for medical devices or animal health. Next. Okay, let's talk about the AI. The AI that we use are proprietary to Biologit. It's a machine learning and a combination of NLP models. And what we do is to try to find any conversations within the text and on the unstructured text where there could be a possible adverse event. It's not defined, yes, it is an adverse event, but is a possibility of implicitly or explicitly having an adverse event, or a new safety information. So the searches can be used for ICSR collection, but can also be used for new safety information to go into signal detection or into any aggregate report, for example, into the DSUR or the PBER. It can also be used for that. So you start with a vast number of hits. So the number of hits that you get can be the same that you're getting today. You can use the same search strategy that you use today with the name of the product plus the synonyms and adverse event terms, for example, but you can also use the name of the product and synonyms and only use the AI filters to help you to filter down the number of important articles that you want to review. So the second bar, the bar in the middle, it will be anything. So the top one will be anything that has the product name involved or any of the synonyms of the product name. And the one in the middle, it would be anything that has a suspected safety events for any, for signal detection, for aggregate reporting. And the last smaller part of it is anything that you also have an identifiable patient. For example, you can see if it's a case report, if it is pregnancy, elderly, and so on. That will be the articles that are most likely to be the ones that you want to concentrate on. So you can automate and filter all of those steps to help you to have to review a much smaller number of articles compared to the very top, where you would be looking at, let's say, 1,000, and then in the middle you would be looking at 500. But at the end, the most important articles that you need to look at will be at the 50. Let's say use the 50 number at the very bottom, and these are the ones that you want to concentrate on. So it is very flexible. The models are monitored, and we do the full compliance with the models ourselves. So they are not being trained by client data, and they also not being trained by information that is coming from clients. We are responsible for the training and maintenance of the models. So it is very repetitive. So every time somebody runs a search, you will get the same time of results, because it is an extraction of safety information from the text, and that is a machine learning model. So it is repetitive, and there's no risk of hallucinations, for example, because it's not a large language model. There's also flexible levels of supervision, so the human in the loop to human supervised in the loop. So you can automate a lot of the steps depending on your appetite for the use of AI. And also, like, you will need to have conversations, obviously, with your quality team, but also within your internal teams to see how comfortable and how much AI is the company willing to use. But you could start with just minimizing the reviews by using some automation, but also QCing some of the results afterwards. It doesn't need to be 100%, but you could QC a portion of it and meet the guidance of the QC methodology. And also, we are meeting industry standards of AI validation and governance, and that is very important, because we want to make sure that what we're doing is in line with the regulations. That's why I had the second slide up front, and what is the expectations from the regulators, pharma, CROs, and so on the development and implementation of AI models. Next. And I give it I'm heading over to you, Bruno.
Okay, Nicole. Let me bring back my camera real quick. Good day, everybody. Thank you for joining today. I'm Bruno Hanna. I'm CTO of Biologit. I run product engineering here. I'm co-founder of the company with Nicole as well. And I'd like to walk through a few of the major updates that we rolled out on the platform in the last quarter, so maybe a little bit more, and talk about the future, and then go over into the platform and do a quick demo. As a reminder, please send your questions to the Q&A, and we'll get back to them for a discussion at the end. So the first thing to call out is our local literature offer that Nicole already mentioned. This is an add-on product that natively integrates with the rest of the platform. And if you think about local literature processes today, for anyone involved in this, it is a very manual process in that, because most of this literature is not indexed anywhere, it requires this sort of one journal at a time assessment, where you will go to a journal website and look for articles there, then move on to the next one, and maybe receive some publications in print and review the scanned copies and so on. So it definitely doesn't scale. And the universal journals in the local literature world can be very, very vast as well. So how we solve this problem is Biologit will become responsible for ingestion, data digitalization, and all the oversight that goes with the local literature requirements, whatever they are, if they're websites, if they're newsletters, if they're in print. Then we'll make them searchable inside the database, which means the user experience in the platform is the same as a global literature search. You write a query string, the query string runs, presents results that match what the user is looking for. And basically that's it. And then the assessment continues from there. So it's a lot more streamlined than having to do this by hand. All the governance and oversight is taken care of. The process is a lot more repeatable, because no matter what the source is or what country it is, the assessment workflow can be exactly the same. And it's all documented in the platform. We're going to show this in a bit more details during the demo. But I think the important takeaway from this slide is that all the complexity that's inherent to the local literature problem today becomes our responsibility as a vendor, so that the user experience is of a much higher level of quality inside the platform. Another very important milestone when it comes to our products are the effort that we have been putting on integrations. That there's always a requirement to maybe ingest data, even though we have a pretty vast database and many of our clients use it today for global and local. There could be scenarios where some external data is needed, and we want to make this process super streamlined as well with integration. So what that means is we can ingest automatically data coming from email alerts or from other sources that we can reach via an API. These results become hits inside the platform that can be assessed just like any other, which is great, because you can also mix and match these results. If there's a third-party source that you like, plus the rest of the results coming from Biologit, that's fine as well. So that's inbound data. As for outgoing data, the challenges are more or less similar. And again, many of our clients that they are pursuing the technology vision of touchless PV, where things are very streamlined and go across from one system to the next with minimal supervision. In order to facilitate that, that means we need to integrate across the spectrum of systems very seamlessly as well. So when it comes to talking to safety systems, Biologit can send its data downstream, either through very well-known integration channels like E2B, which I'm sure many of you are familiar with, or through more direct methods that integrate with APIs that other third-party systems can expose. So all this means that literature becomes very integral to the entire flow of PV without a lot of handholding from one system to the next. I did not mention a whole lot of different things that are on our release notes. So we do have a pretty fast release cycle, which means we like to deliver benefits to our clients on a regular cadence. So enhancements to the platform, new reports, more flexible workflows. This is all something that we ship fairly regularly as well. Here are some fun numbers coming from the platform today. We're processing about 3 million hits on an annualized basis. I can confidently say, from a year ago, this number more than doubled. We expect this to continue to grow, which speaks to the scale of the platform. There are today more than 2,000 products under surveillance in Biologit. Our database has over 88 million citations. It grows at a rate of about 45,000 citations every day. We have coverage of over 100,000 journals from pretty much all over the world. So that's pretty significant for a global literature requirement. We are more than compliant. We do include PubMed and Medline, which are gold standards in the industry, and we complement that with many resources to the point where our global literature can help many of our clients solve their local literature needs as well. But in addition to that, today we're also capturing data from up to 3,500 or so non-indexed local literature sources. So these are journals that maybe have exposed their publications on the website, but they're not really reliably indexed in any database anywhere. Or there could be newsletters that are not even journals or websites of interest and that sort of thing. So today, we already have a pretty significant coverage there, representing more than 140 countries, and that continues to grow. This is about what's coming up next. What the general theme here of our roadmap is we want to continue to be the technology leaders in the space. We know that the key to our success is to be focused on the literature. This is the problem that we chose to be really good at, and it's reflected on our platform. So we also know that from our many conversations with our users that local literature in PV means productivity is key. So there will be more and more investments in making the platform the PV assessor's best friend in terms of getting things done fast and getting things done reliably. So here's some examples of what we're doing in terms of productivity. We want to convert things that take multiple clicks to maybe a single click or no click at all. Really support users in completing their day-to-day workflow faster. We're also going to invest in agentic technology to solve these more challenging problems inside PV. So, full text discovery might be one. How to reliably extract data from the contents of literature data to speed up assessment, to speed up data entry, and so on. So these are problems that we're investigating, and we want to roll out some nice solutions this year. And then finally, we'll continue to invest on enterprise features, be it integration, security, so that we can continue to be part of the big picture on solving the challenges of PV. In addition to that, we'll also make investments in delivering more comprehensive multi-tenancy support, which is something many of our clients are very interested in. Okay, that's it from me in terms of the presentation. we can jump on the platform now for a quick demo of some of the things that we discussed. The platform now. So I'm logged on to Biologit here. So this is the landing page. As you can see, it's a web-enabled SaaS that has a very familiar menu-driven kind of approach. What I'm going to show is some results that are already created on the platform to illustrate some of the AI capabilities and how they can be put to good use when it comes to productivity, as well as local literature. So when I log on as an assessor, I would navigate to this review section here to see what searches have run on the system and have been assigned to me and their pending assessment. So in this demo, there's a small portfolio of products, including local literature, and maybe a dedicated search just for health authority websites. So these searches are already run on the system. They can run on a schedule. And when they're ready, they get presented to me. Now I can complete my assessments. I'll start with a particular product here that's representative of how global searches are organized. If I click on it, I'll see a list of citations that were retrieved by my search. And they appear in this tabbed view here. So in queue is the work for me to complete assessments, whereas reviewed, the assessment already happened or it has been automated. So a number of actions already were performed by the system before these results are even presented to me. An important one is duplicate detection. And we can see here in the duplicate tab that out of these 226 results, 71 of them were flagged as duplicates. And the duplicates here can be identified because of the contents or because they appeared on a previous search for this product. That there are lots of different mechanisms for us to detect them. But they get automatically sidelined. That means I don't need to worry about those anymore. They're still visible, as with any automated decision on the system, which means I can check them, I can QC them if I wish. The other action that happens as these results got presented to me are the tags. So the tags correspond to our machine learning models, and they're here to help answer a particular question that's relevant for pharmacovigilance assessments. So for example, our main model, the suspected adverse model, which is this blue badge here, means that the system scanned the contents of the title and abstract and identified a potential safety event might be discussed here. This is by definition and by design a pretty broad category, because when we thought about these machine learning models, we really didn't want to create something that would introduce the risk of missing important safety events. So they're designed to be conservative. But even so, as you can see here, they can still deliver a pretty significant productivity boost. In this example, about half of the hits that were returned, they have the flag, which means the other half does not. And so that creates the opportunity for us to think about more streamlined workflows to cover the non-relevant or the ones that are very unlikely to be relevant for our assessment. And we can also automate them, which is how I configure this demo here. If I go on the review tab, I can select the citation screened by the automated user, which is this one. So there's a number of citations here that were pre-screened by the system because they don't contain a particular AI prediction or AI tag that's relevant to me. Again, they're still visible here, which means whatever oversight methodology we want to apply on top of this, it's possible because I can QC them, I can sample QC them, and so on. But that's another mechanism by which the technology can deliver productivity here. We see another 52 citations have been automated away in the search results, leaving me with a more focused data set to review as an assessor. The last thing I wanted to show here is how an assessment gets carried out on the platform as well. So if I go, I'm just going to show you a citation that has been completed already, so we can see all the aspects of it. So here's how the citation gets presented to the user. There's going to be a number of highlighters to help me focus my assessment. For example, the red highlight is my product. Here's an identifiable patient. And the sections in yellow are the areas where the content is most likely be discussing a safety event. So I can use those if I want. I can also switch them off. And to continue my assessment, because this looks like it's an important safety event, I can procure the full text. For that, the system will present me additional metadata that will help me navigate to the journal, get the full text if needed. I mentioned before that we're focusing on agentic productivity here, so this is a problem that interests us very much in terms of integrating full text procurement with platforms that exist today, as well as identifying ways for the system to find those ourselves. Once the PDF is identified, I can upload it back into the system. Like basically any evidence that's relevant to my assessment goes here. And now it becomes visible from the platform, and it's going to stay here for future rounds of assessments like QC or safety review and so on. And then finally, I can complete my assessment, which is a fully configurable workflow that we see on the right-hand side here. This is a very simple valid invalid ICSR, but anything can be configured here to reflect the exact steps of your workflow. Because this is going to be a case, I can already enter details corresponding to E2B fields in this E2B section here, where we see patient, drug, the particular event, and so on. So these are all mapped into E2B fields. And now once I save my decision, when everything is done, either the system can allow me to get these XMLs to send to the case processing team. I can get other formats as well, like a narrative or a zip file. But also coming back to integrations, once I save my decision here, that can trigger an action on the system to automatically go and talk to an API to deliver these E2B file to a downstream system, and then processing continues there. In other words, there's nothing for the user to do in this case. I just save, and the system will take care of the rest. Okay, so that's the productivity and with an example of global literature. Lastly, I just wanted to show how local literature looks from inside the system. Here is an example of a local literature search for Indonesia. And unsurprisingly, it looks very much like global, which was our objective. So what I'm seeing here is a list of citations that were retrieved for the journals that I'm interested in, that are matching the terms that I'm looking for. So this is synonyms of ibuprofen. And now I can continue my assessment following my workflow, pretty much as if I was screening in any other set of results coming from a database. So that's a pretty seamless experience. The only possible difference here is content that you can see may come on the original language, but we support machine translation out of the box, as well as integration with the translation systems that are preferred by our users. Just to give you a sense of how this looks like from inside the platform. So this article is in Indonesian. It was a match. maybe the first thing I would like to do is request an abstract translation, which will bring me the English version of the content. And I can continue on and do the same for the full text of the article, the original, and we can request a layout preserving version of that, which is in English with machine translation. So all these options are available to users to kind of speed up the process. Okay, great. I know we're almost at time, and I'd like to leave some time for questions. I think that concludes the demo on my side. Thank you very much for joining today. Maybe we can open up for questions.
Bruno, there's some questions here. Are the flagged duplicates categorized, already identified literature references, potential duplicates based on content, in order to enable people wishing to QC them to target their QC? So it's a question on the deduplication. What is the process of deduplication?
So any duplicates will appear in a particular area of the system. So I go into the results and I go into the duplicates tab. So these are automated exclusions with the justification being they are a duplicate. The system will tell me some information about help QC. So when I'm inside the duplicate, it's going to show me why. So why is this a duplicate? And who is the other article, which I can navigate to. So in this example, this was an article this was the same article that appeared twice, once from DOAJ, which is a database that we ingest, and also in PubMed. So the reasons for that are here, and it's visible to the user so they can review and QC.
And then Bruno, along the lines with that, this one example is because it was already in another database. But what if there's a question that we get a lot? So what the deduplication, for example, if an article gets reviewed and then a new article gets published with very little to no information. For example, if it gets sent back, if it gets published again, but just with a EPUB, like before publication date, and then afterwards with no changes to the text, that also gets picked up here, doesn't it?
Yeah. Either it will be picked up or it might be referenced back by the DOI. It depends on how it will appear in the second time.
We do have a hand up, and we have a couple of other questions. I think the question is from Stanislav. I can't say the name properly, but please ask the question. Okay. I think you need to go on mute. I can't.
Hello, can you hear me?
Yes, I can hear you now.
Yeah, thank you for the presentation and for presenting the demo. I just want to ask, so if I understood correctly, the first assessment is like flagging the certain keywords in an article. How technically it is done? Is it like that the machine learning just identifies these keywords and it is trained on these keywords? It's just some text embeddings, or how is like the first assessment by machine learning or AI done?
So maybe to be clear, when we're searching a database, that step doesn't use any AI. It's using regular query strings with booleans and wildcards and things like that. So these this is the method that's more familiar to the industry as well as more auditable. So that stays the same. Then what you see here on the right-hand side is predictions. The technique that we use to produce them, it varies from one method to the other. Generally, it will be a combination of neural machine learning, where we collated the data and trained the models ourselves. That or combined with one or more natural language processing techniques. So yes, it's a neural model. It does use embeddings as well. I think you asked that. There's some reference material that we put out publicly that describes these models in more detail. Well, one of the things that as a company we double down when it comes to machine learning is being as transparent as we can in terms of how these models were put together. What do we do by way of governance, and how is the training process, and how is the testing and validation process? So this is all very well documented. Something we talk about a lot when we go on industry forums, and there's a good deal of available documentation today.
Thank you.
I have another question here. So does the AI tool assess based only on abstract? If so, how was the causality determined for paclitaxel unstructured in the demo? I can answer both of them if you want me to answer. It is based on the abstract title and metadata that is presented. Not all full text are available. The free text is available, the free full text, if it is open source, but if it's not open source, it's not freely available. And to train models, we trained on the abstract metadata. So the keywords that you see there have been used, also the title and abstract. And
so just to complement this question here, the objectives of these models are to narrow down the universe of articles that are relevant. But you mentioned causality, which is the trickiest and most complex part of the assessment, right? So that's not done by the platform. The objective here is to present the user with the most relevant ones, and maybe automate away the non-relevant ones, so that the set of results for assessment is more narrow. The other reason for why causality is tricky is because causality may be only talked about in the full text of an article, which we don't necessarily have access to. So what we present here are the candidates. You can think of them as the most likely candidates for review, which then can proceed with the assessments in a more granular level.
Thank you, Bruno. I think we've answered that question. So no causality is determined by the system. So, but the reviewer will review less articles. And for example, the combination of tags, I don't think you went through that, but you can use a combination of tags to make sure that you will be reviewing the ones that are most likely to have an adverse event. So for example, if you have your target product, if you have a suspected adverse event, is a suspected, is not identified adverse event, is a suspected adverse event in the patient. So you have the 4 criteria, and now the reviewer will have to do the assessment if it is caused by the product or not. It also highlights the concomitant medications to help you find it within the text, and that will help you to say, is this, is my product the target that has caused the adverse event, or is it a concomitant medication that was in the text, for example. I also have another question here. Can full text article be ordered directly via API from the preferred full text provider and loaded into the system automatically? Do you want me to answer that?
The answer is yes. we're happy to integrate with any vendor that provides some sort of programmatic access for full text procurements. There are also many full text procurements in the industry today that do a pretty good job at scanning a web page and looking for relevant information to point users to the right article. And that's great because we provide this metadata as well. So for example, from the DOI here, many of these browser-based tools can pick that up and already provide the user with an answer. But yeah, we can integrate as well through APIs.
Yes, and the API will be an API of your choice that it can be configured into the system. And then I have another question. Is duplicate check limited to data in Biologit or against the safety database too? I can answer partially and then I'll let you answer. So it depends. If you've been using our platform for many years, all of the information, any of the literature reviews that you have done will be in the system, and they will be deduplicated based on all of the information that is in the system. If you are only starting to use the platform now and the information is in the safety database because you've done the search in another either manual or so on, at this point in time, which is at this point in time, but that is maybe something that is interesting for us to investigate, we could talk directly to the safety database, but I think that is not an integration that is there today to search the safety database to see if the same article is there. But the answer is, if, for example, you've been using the platform for a while, if anything that was published 3 years ago and is published today, again, you will be able to see that is a duplicate. Let me see, we have some more questions. Are you actively searching for local medical journals available and possible updates to the list, such as termination in the country, or is an activity that needs to be supplied by the client? And I think that the answer for that is usually we work with clients that will provide us the list of local journals that they are interested, and then we will configure the system to the local sources that they want for individual countries. That is how we usually work. But because we have a lot of knowledge and we have most of the sources, we can also suggest what journals, depending on the products that you have, and it also depends on the product. So if you have an oncology product, you're probably not doing searches on I don't know, sports medicine or something like that. So it depends. The list of journals will depend on the products that you have, but we can definitely help and support in any way with the knowledge that we have internally. I have another question. May I see the different options available in Review Decision tab? So it's a valid ICSR, and this is configurable. You can configure it to be any decision that you wish. So this is a very simplified version of the possible decisions. So it's a valid ICSR confirmed, invalid ICSR confirmed, signal, aggregate reporting. This could be two separate entries as well, and pending translation, pending full text. And that is why it's important to have the translation integrated into the system, so you don't need to use that option, and the full text as well. If it's fully integrated with the system, there's no waiting time. You can download the full text straight away and download the translated article, and you can finalize your review.
Yeah, this menu can support multi-stage workflows as well, which we see quite often. So there are assessor level options, and once the assessor saves a decision, then it becomes a menu for the QCer or for the safety assessor with different options. So that's supported as well.
But this is configurable. So it depends on your own workflow. If you have a different workflow where it's giving different options that can be implemented. For example, for clinical safety, it might be very different. If it's used for toxicology, it might be different decisions as well. I think we only have 2 minutes. I have one very long question here. I will ask that question and then we can answer any questions that have come to us. We see a way of getting back to you with answers for them. For the questions I have here is, how exactly are you bridging the gap between high performance black box models and the strict transparency required for JVP audits? How is Biologit architecting its NLP pipeline to ensure that automated screening decisions, especially for unstructured local literature, are fully explainable and audit ready when regulator asks exactly why a specific article was dismissed?
I was looking at the as well. It's a great question. So we build our AI technology fundamentally around regulatory requirements. So we watch this space very, very, very closely. We also maintain a dialogue with regulatory authorities through inspector forums and direct interactions with the likes of EMA when that's available. So all this is to say is when we build the AI software development lifecycle on our side, we had all these considerations in mind. One thing that we're also monitoring very closely is what's coming out now on Annex 22 for the EMA, where these kind of guidelines and expectations are being refined now. And it goes back to one thing that Nicole said at the start, that there needs to be a documented and justified risk-based approach on the design of the system as well as on its use. So we're very involved on active implementations that we have today to make better use or to take more advantage of AI where those topics come up. So the level of justification can be statistical, but it needs to be very well justified, and it also needs to be very well justified how the risk is going to be managed. But fortunately for us, number one, we're very transparent. We can show in great level of detail how we build those models, and in addition to that, we can collaborate with our clients based on the data that they're observing, right? Are the assessments that they're making inside the platform corresponding to the AI prediction? So that's very useful input to build in a risk management plan around AI automation. So that's basically the approach that we take. I hope that helps answer your question.
And I just want to add that because within the platform you can see all of the results, you can see all of the results from where they're coming from. For example, are they coming from a local source? Are they coming from a regulatory authority? Are they coming from what database they're coming from? What sources they're coming from? You can see that. So it's fully transparent within the workflows in the platform for companies to be able to audit the results, and that companies will then make a decision on what they need to audit based on a risk-based approach. So nothing is hidden away. So for example, if the AI is making assessments of this is not relevant, for example, you can always go back to that. You have full access to what the AI has deemed not relevant and audit that. So the black box is not really there because there is a transparency on the outputs. I don't know if I hope I have answered.
There's also greater acceptance of statistical machine learning models now. If you read the most recent versions of regulations that are coming out in drafts, and they're setting expectations around it, where they seem to be drawing the line is on large language models. That one is a universe that the regulators are still thinking about. But when we talk about quote unquote classical machine learning, where you have control of the training data, have control of the model, and the test results. Things are getting things have a lot more clarity now.
Okay, so I think we're 2 minutes late, but thank you so much for everybody who joined the webinar and listened to us. We are very open to have further conversations with any of you. Our inboxes are always open. I have you have my email. I think it's not. You probably have I don't know if you have my email, my LinkedIn. I'm always open to have conversations with anybody. Rajat is also asking if you have any additional questions to put in the feedback link that he will share. So that will be helpful. We can get back to you. But thank you so much.
Thank you very much for joining today.
Thank you.
Have a good weekend.